Hello everyone, in this video we'll be discussing about non-cyclic photophosphorylation. So let's start the discussion with the word photophosphorylation. What do we mean by this word, this complex term? So we will break this term into two parts. One will be photo. Photo means light and phosphorylation means adding phosphate to the reaction. Now what happens in the presence of light? ADP molecule, which is adenosine diphosphate, will accept one inorganic phosphate and convert into ATP, that is adenosine triphosphate, the energy currency of a cell. So if this process occurs in the presence of light, we call it photophosphorylation. Phosphorylation means adding phosphate to a substrate. So we can define it as synthesis of ATP from ADP and inorganic phosphate in the presence of light. Now let's see the story. We know that the light absorption in a plant occurs by light harvesting complex, and we have two types of light harvesting complex. One is photosystem 1 and one is photosystem 2. When photosystem 1 and photosystem 2 work together, they achieve more. That is, when they work together, photosystem 2 will function first and will send its electron to photosystem 1. Then why they are named such way? They are named according to the sequence of their discoveries. You need to remember this. So when both of these photosystems work in series, it is called as non-cyclic photophosphorylation. This is the topic we are discussing right now. And as I told you, when these two photosystems work together, they achieve more. They will synthesize both ATP and NADPH and H+. But when photosystem says that I want to function alone because I am the topper, as we have already discussed, photosystem 1 has an absorption peak of 700 nanometer, whereas the photosystem 2, which is the runner-up in our class, has an absorption peak at 680 nanometer. Now when photosystem 1 functions alone, will achieve less, as it is always said that growth with friends is exponential. So photosystem 1 when functions alone is called as cyclic photophosphorylation, and here only ATP is produced. NADPH plus H plus is not produced. Now let us see this reaction in detail. This is the NCERT diagram, and here we'll be discussing the steps of non-cyclic photophosphorylation. Why I call this non-cyclic photophosphorylation? Just because I can see both photosystem 2 and photosystem 1 are working together, and when they function together, it is called as non-cyclic. When photosystem 1 functions alone, it is called as cyclic photophosphorylation. Now let's start the discussion. So since it is light harvesting complex of photosystem 2, I'll mark it as PS2, and this is the light harvesting complex of photosystem 1, I'll mark it as PS1. When PS2, the light falls on the reaction center of this light harvesting complex, the reaction center chlorophyll A molecule absorbs the light and excites the electron. These excited electrons will move through antenna and will move to a electron acceptor which has a higher redox potential. So here we have that electron acceptor. Now this electron acceptor will send these electrons in the downward directions of redox potential to the light harvesting complex of photosystem 1, and this journey of electrons occurs through electron transport system. And my friends, this electron transport system is made up of cytochrome B and F, best friends. This denotes the friendship of both photosystem 2 and photosystem 1. So when the electron passes through electron transport system, there occurs the formation of ATP, that is adenosine triphosphate. Now these electrons are handed over to the reaction center of light of photosystem 1. Now the light harvesting the reaction center of photosystem 1 will excite these electrons further and will send it to an electron acceptor which has yet higher potential than the previous electron acceptor, higher redox potential. But this electron acceptor will betray everyone in this story. Photosystem 2 is still waiting for its electron, which he has sent to Photosystem 1. Now Photosystem 1 says that my friend Photosystem 2 requires his electron back. But this electron acceptor says, I'll not return it back because I'll send it to a drug mafia named NADP+. And this will convert into NADPH after receiving the electron. Now this Photosystem 2 is crying, hey Photosystem 1, you took my electron and have never returned it back. So now how will I deal with you? So now comes a generous man, which is water in the story. Water says, I'll destroy myself to produce electrons for you guys. So Photosystem 2 stops crying and laughs again. Because now the water molecule is giving its electron, protons and oxygen atom. When two molecules of water will be there, will be having oxygen molecule, and this will be released as oxygen gas in the surrounding. So this is the whole setup of non-cyclic photophosphorylation. Why it is called as non-cyclic? Because electron once gone will never come back. But in cyclic photophosphorylation, the electron keeps on cycling in the same electron transport system and keeps on producing ATP. So in non-cyclic photophosphorylation, this drug mafia, which is NADP+, is unique. And therefore, when I told you, when these two friends will function together, they will achieve more. And through this electron transport system, you can synthesize ATP. But for synthesizing NADPH, you need them both working together and a drug mafia named NADP+. So these are the steps of non-cyclic photophosphorylation. Let us discuss individually. What is the first step? So first step is the reaction center, which is made up of chlorophyll A molecule of photosystem 2. It will absorb the light and excite the electron. So we are talking about this stage right now. Light is falling on this light harvesting complex and it is exciting the electron. Now what is the stage step 2? The excited electrons would be picked up by the electron acceptor. So now we have reached the story up to here. So we have an electron acceptor which is accepting the electrons. This is step 2. Now what is the step 3 of this non-cyclic photophosphorylation? These electrons now pass through electron transport system consisting of cytochromes B and F particularly. And this movement of electrons downhill in terms of redox potential scale. So this is the step 3 which I am marking right now. That is movement of these electrons downhill through electron transport system. Now let's talk about step 4. These electrons will be transferred to photosystem 1 and the reaction center of photosystem 1 will absorb light and excite the electrons again. For exciting the electrons, these require light energy, which is marked by this yellow colored arrows in this diagram. So now we have reached this level where the reaction center of photosystem 1 is absorbing light to excite the electron. Now let's talk about the next step, 5th step. These excited electrons are transferred to another electron acceptor which has higher redox potential as compared to the previous electron acceptor. And the last step, these electrons are moved downhill to a molecule of NADP+, which I am calling a drug mafia. And addition of these electrons to NADP+ will lead to the formation of NADPH plus H plus molecule. Therefore, at the end, we have both ATP and NADPH plus H plus being formed in non-cyclic photophosphorylation. We call it non-cyclic because electron once gone will never return back. Now let's talk about Z scheme. We have already discussed in light reaction that this characteristic arrangement of these pigments, which we can see, is in the shape of N. But when we see this N from this direction, we could easily form a letter Z. Therefore, this whole scheme of pigments arranged according to their redox potential scale is called as Z scheme. So that only we are discussing right now. The whole scheme of transfer of electron is called as Z scheme due to characteristic shape formed when all the carriers are placed in sequence of redox potential scale. So these three statements should be on tips to all of you, that what is Z scheme. The whole scheme of transfer of electron is called as Z scheme. Why it is called Z scheme only? Because of the characteristic shape formed. And why this shape is formed? Just because all the carriers are placed in the sequence of redox potential scale. And with this, we have completed the discussion of non-cyclic photophosphorylation, when both the friends photosystem 2 and photosystem 1 are working together in series to produce ATP and NADPH both. So with this, we have completed the discussion. Hope you like this video. Do check my other content also. And I upload these notes on my Telegram channel. Do visit my channel also.